to develop a testing approach that can assist growers and agronomists to make safer planting decisions after having applied imazapic and diuron. So many of you will be aware that there's been an increasing use in pre-emergent herbicides, maybe in response to herbicide resistance in other key herbicide groups. So two of the most commonly used herbicides are imazapic and diuron. The difficulty is that for many growers, there's a bit of a lack of clarity about when it's safe to replant, even when they've adhered to the product labels. I've taken these comments directly from an imazapic product label, so you can take a moment to read through this slide. Okay, and similarly for diuron, another little snip from the label there. So just take a little moment to read that.
Was there anything in particular we needed to focus on, Annie?
All right, so I'll just go back. So just have a little look there for the imazapic one. So basically you can see there that, that it's always possible, according to the label, that you could have rotational crop injury. And if you look down, if, you know, for the comments upon following use in summer fallow, there's, you know, a lot of sort of terms and conditions there. And unless you're using sort of crops with clear field technology or maybe planting something like chickpea or mung bean, you know, it's a pretty complex situation in terms of rainfall, etc., to know whether or not it's going to actually be safe to replant. And so again, if you look at the diuron product label, again, there's a lot of terms and conditions there. There's a lot of variables, so that even with the best of intentions following the herbicide labels, there's so many factors that can influence how quickly residues will break down, and decisions about replanting are not always going to be straightforward. So in essence, growers need to understand whether or not it's going to be safe to replant subsequent crops. It is possible to currently determine total herbicide levels in soil, but this isn't really meaningful because depending on the soil type, only a fraction of the total is going to be available to the crop. And there's no current test to determine the bioavailable herbicide concentration in the soil for these herbicides. So what that means is how much of the total can the crops actually access, and that's going to vary between soils, and currently there is no test to know what the bioavailable portion is. And furthermore, even if we did know this, we don't actually have any information to relate back bioavailable levels back to the crop species to know whether it is safe to plant a given crop. So in this research, we're aiming to develop these three components, and the idea is this will help to develop a future decision support tool for growers. So we at DAF are working on identifying the crop toxicity thresholds and also a sampling protocol for collectors such as farmers and agronomists once there is a test to determine bioavailable levels. And our New South Wales DPI colleagues are developing the testing approach for determining the bioavailable herbicide levels, and that's going to hopefully work across multiple soil types. So this is a bit of a snapshot so far of what New South Wales DPI have been achieving. So we've helped them, and we've collected quite a few soil samples. The soil samples have come across key areas of the northern cropping region, and the idea is that this information feeds into the soil test so that when the test is developed, it should work across a range of key soil characteristics. So there's a pretty good spread there you can see in terms of organic carbon and the cation exchange capacity. So we've managed to get a pretty comprehensive suite of soils together to inform that soils test. I'm moving on to the sand bioassays bit, which is the bit that we've been concentrating on at DAF. So we've been preparing dose response bioassays, presently using sand, to identify the amounts that can impact crops at maximum bioavailability, and we'll then extend these results into bioassays with real soil types to see how this bioavailability changes in contrasting soil types. So this is our 4-day quick test that we use for imazapic. This is quite a nice rapid test. You can see that we mix in quite discrete amounts so that helps us to be nice and accurate with our rates. So these seedlings are grown in controlled climate so that the conditions are good for the crop growth. And what we do is we measure, in this test, we measure the root length, and we measure it quite accurately to compare changes in root growth across rates. So this is a photo for sorghum, which has been one of the crops we found to be, most sensitive so far to imazapic. You can see there that there is a change in shoot length, but really it's root length that seems to be the key indicator of seedling response to imazapic. So in this one, we've got the black arrow there, and that's showing 0.03, and that's milligrams per kilograms, which is equivalent to the target rate for imazapic in fallow use situations. So this is the dose response curve for that experiment that I've just shown you. You can see there in the black, again, that's the label rate based on the lower label use for fallow weed control in the product. The commercial product that we're using is called Impose, and that red arrow there indicates where the significant change has occurred, and you can see that has occurred before, at quite a lower dose than with the than where you see the black arrow shown. So this shows that even given considerable herbicide breakdown, crop injury could still occur in sorghum in maybe some of the lighter soils. I'm moving on now to diuron. The rapid test isn't appropriate for this herbicide, so we grew the seedlings in larger pots called forestry tubes, but they're still quite small so that the mixing of the sand and the herbicide is easier and still quite accurate. In these forestry tube tests, we grow the plants for 21 days in controlled climate growth rooms, and after 21 days we remove the seedling from the sand, we wash them and weigh them, and we collect data on fresh and dried weight and also shoot length. So this photo shows barley at 21 days, and even from this photo you can see that the damage occurs before the target rate, which is shown with the black arrow. This rate is based on use in cotton because this is quite a robust rate, and in our region it's not uncommon for people to replant cotton areas to grain crops. And also, unlike imazapic, fallow weed control is not currently permitted using diuron in most Australian states aside from South Australia, and in South Australia the fallow rate is quite low. So again, we've got the dose response for that experiment. Barley has been one of our most sensitive crops so far, but we haven't sort of done as much work in diuron yet. So again, if you look at this, you can see that herbicide concentration occurs, the damage occurs at that red arrow. So that's quite a bit lower than the black arrow, which indicates that even after a reasonable amount of time for herbicide breakdown, you might not be out of the woods for herbicide injury. And even using the fallow rate permitted in South Australia, which would be equivalent to about 0.1546 grams per kilo, you can see that in lighter soils that could reduce barley growth significantly. So in terms of the tolerance that we've sort of seen so far we've seen that in imazapic, the sorghum is our most susceptible, and there is pretty strong indications of natural tolerance indicated in the pulse crops, chickpea and mung bean. In diuron, so far the wheat and barley seem most susceptible, but we're not as far along with that work. So from the label for imazapic that we looked at earlier, unless you're using certain lines of wheat or maize varieties with the Clearfield technology, you will have to you would need to be careful with crops like sorghum, and this reinforces our results. So it sort of matches up with the label pretty well. Also from the label, chickpea and mung bean have less special conditions to meet for replanting, so that is also validation for our results. And in diuron, the label recommends that you do not, for cotton, so if you apply the rates, which is what we're sort of basing for the cotton situation, then the label states to not replant treated areas to any crop within 1 year of last application, as injury may result, except for cotton, corn or grain sorghum. So this kind of also validates our results, because our least susceptible ones so far for diuron do seem to be the sorghum and maize, so which would be the safer replanting options. So this sort of just shows that if farmers do know the susceptibility thresholds for certain crops, then even if they've got high residue levels as indicated from a test, there might still be some good crop options for them, and some that would not be a good idea to plant. So going forward, there's more work to be done with bioassays, because we need to do this same work in actual soils. So we've collected some quite contrasting soils, and we'll be doing the same bioassay work with them. And with those soils, once the test has been done, the herbicide residues will be extracted and quantified by our colleagues at New South Wales DPI. And the guys at New South Wales DPI will be doing some testing involving radio-labelled diuron and unlabelled imazapic to derive sorption coefficients for the contrasting soil types. And the sorption coefficients will then be used to convert measures of total herbicide residues in soils to bioavailable herbicide residue levels. And the idea is that machine learning approaches will be used to relate the MIR spectra to sorption coefficients. And the benefit of MIR is that once a suitable calibration has been performed, analysis of new samples is very quick and inexpensive. And moreover, the MIR spectrum is already being used by commercial laboratories for soil analysis. So just to recap, this figure was put together by Mick Rose and is a nice summary of how the different components of the work feed into each other to enable the development of a tool to assist growers and agronomists to understand plant-back risks for certain crops based on soil tests that capture bioavailable herbicide residue levels in their soil. So this test has real potential to help reduce the confusion associated with plant-back decisions, and hopefully will reduce crop losses due to herbicide damage in situations where herbicide levels remain high for whatever reason, even after following the herbicide messages on the labels. So this approach is also quite useful if going forward we want to adapt this approach for similar work with herbicides that pose a risk due to herbicide carryover. And finally, I'd just like to thank GRDC and all the people who have been involved in this research. So this should have, I think I've gone through this pretty quickly. So, I think we can probably ask questions now. Is that right, Michael?
Yeah, no, that's right, Annie. So I've suggested that people can enter their questions via the chat option.
Okay.
If you're able to have a look on there, you've got one there ready waiting for you from Ali.
Okay. All right, so let's have a look. Okay. All right, so we aim for 80% of water holding capacity for the soil moisture contents, which is really quite generous for the plants. So that, you know, we mix our concentrations into the water to the amount that would achieve 80% water holding capacity. Does that answer your question, Ali? Okay, good. Anybody else? Yeah, I think that's a good question. So the question from Gulshan is the diuron breakdown related to temperature because it's susceptible to winter crops. I don't really know enough to answer that properly, but the herbicide label does specify different periods for replanting for summer and winter crops. So it is possible that it's related to temperature, but I think it is also just to allow that amount of time for the herbicide to break down.
Annie, Mark Congreve here. Do you want me to jump in at this point?
Yes, please, Mark. Go ahead.
So, yeah, look, temperature is a factor in speed of breakdown. We need temperature for microbes to break the herbicides down, but moisture is probably more important than temperature. So if you're in the middle of winter, you've got low temperature, and even with moisture, you won't get a lot of microbial activity. In summer, you've got plenty of activity going on from both, having moisture and temperature. But conversely, in summer, if you don't have the moisture, then you won't have any breakdown going on. So it's a combination of both temperature and moisture. The other comment I'd make also, while I missed the first part of the presentation, I had some difficulties logging onto the system. I think I stuffed it up. But anyway, I got in. When you start doing the field correlations, particularly with imazapic versus diuron, need to understand the mobility of those two herbicides within the profile. Imazapic's a very soluble herbicide and it will move down the profile with rainfall. So what you've been doing in the pots, you're basically placing the seedlings in direct contact with the herbicide. So that's going to be a worst case scenario. Diuron's going to stay much closer to the soil surface. And so, one of the, I guess, strategies you can use with diuron is if you're planting a bit deeper, you may keep the seed away from the herbicide. Whereas imazapic, you're not going to be able to do that. So it's going to get more complicated when you get out in the field because you've got where is the herbicide in the profile. You can't just assume that it's going to be the same between different herbicides with different mobility.
Yeah, so that's a good point. In this project, there's no scope for actual field testing. So right. The work will all be done in pots, and in the pots we do make very, very sure to make sure that the herbicide stays in the sand where it should be or in the soil where it should be. We don't allow any watering that would permit runoff at all. So we water very carefully. We water by weight, and at the beginning we weigh everything at the 80% water holding capacity, and then over the period that we're growing them, we only add water to top it back up to that level, so there's never any water loss.
Yeah, okay. So you're not going to get any loss, but you might find still, depending on the pot size you're using, your diuron staying closer to the top of the pot, the imazapic moving, pretty much dispersing through the whole soil volume with the available soil moisture.
Yeah, so when we mix through the entire soil. So that's why we use the small pots. So we mix individually for each pot, and we mix that soil with the solution really uniformly so that it's throughout the whole lot. And then we place the seed directly into that soil at the planting depth. And because we don't have a lot of watering, because we grow it in that sort of controlled way, there probably won't be as much movement of the herbicide as might occur in a field situation. And we do want it to be like that because we need to understand what can happen in worst case scenarios.
Yeah, that's really good summary. And yeah, so you're getting the worst case scenario where you've got the herbicide directly in contact. When you're starting to analyse in paddock for, when growers sort of ring up and say, I want to do some testing, I sort of say, well, yeah, something like a imazapic, you probably got to do sampling at different depths to actually find where the herbicides ended up. Diuron, you'd be pretty comfortable that a 0 to 5 centimetre sample will probably pick up where the diuron is if it's still in the soil.
That's really good. We might, because part of this project is going to be developing protocols. We've already got, draft protocols together. I might flick them to you at some point, Mark. They're the protocols that we won't be, doing that kind of work now where we are widely testing over a large area, but the idea is that once this test is ready, we will validate it with some results from farm, and we would like to know from you, some little tweaking points for making sure that the samples that are going to be sent to us, do come from those, correct spots in the soil profile. That'd be really good if you wouldn't mind having a look at that.
And particularly with those really mobile herbicides like the Group Bs and the Group Is, picloram, chlorpyrrolate as well. If you've got a hard pan or a significant change in texture at depth, you'll get those mobile herbicides will move down and they'll sit on top of the hard pan. So, that's something that you sort of point out to growers they need to be aware of if they've got soils like that.
Yeah, that's a really good point. So yeah, there's some tweaking probably to be done. I'm taking from that comment, some tweaking for our collection protocol that could help. Yeah, thank you, Mark.
No worries.
Okay, so I'm not seeing any more questions coming through the chat. Was there anything else finally before we move on to Sal's presentation?
I think you might be all good, Annie. Yeah, no, that was good. Well done.
Thank you. Okay, great.
Consider us all giving you a round of applause. All right, thanks, Annie. Okay, Sal, did you want to share your screen now?
Yep, sure, Michael.
Okay, so just, for those who've joined later, Sal is going to give us an update on the cover crops research that's been underway over the last 12 months.
Just 1 second, Michael.
Yep, so your screen sharing is working, Sal?
Working?
It was.
Just dropped.
Oh, so that was it, was it?
It really was.
Okay, now you're good.
Let me just go back. All right, work backwards today, apparently.
Yeah.
Anyway, thanks very much, Michael, for the opportunity to speak today. My presentation will be mainly on the cover crops and mulches component of the US 84 project through GRDC. This is definitely a collaborative presentation. Most of the work that's presented here has been done at Queensland Department of Agriculture with Michael Wedrick and Annie's team, and at Narrabri with Michael Walsh and Assad and his team. So cover crops, by definition, are plants that are established to cover the soil, and rather than using it for harvesting, they're mainly used to improve the soil condition and also to combat weeds. So in essence, the expectation is mainly over the summer fallow season to improve soil infiltration and irrigation efficiency by the provision of micropores, enhance soil cover to reduce soil erosion, therefore conserving soil moisture later on through cover, increasing biodiversity, breaking pest life cycles, and of course reducing the burden of annual weeds, both susceptible and resistant to herbicides. But as you can see from this table here, the agronomic benefits and the ecosystem services provided by these covers changes quite a bit based on the categories they're in. So for instance, grasses are better at nutrient scavenging and also suppressing weeds because they're prolific growers. Then in terms of legumes, they produce nitrogen through atmospheric fixation of nitrogen, and brassicas are very good at suppressing soil pests. So it makes sense to determine the type of cover crops that you need based on your system. And also it also makes sense where you can have a combination of those to have a combined outcome. So the objectives of our project was to mainly identify and characterize weed suppressive cover crop species, whether these are established as monocultures or multi-species mixtures, and that needs to be spanning the whole of GRDC northern region, all the way from Wagga up to Toowoomba, Queensland. So here we look at the different species of cover crops, the density of them, especially in terms of multi-species mixes, the termination time, and therefore its impact on soil moisture. And we did this in 3 sites in Wagga Wagga, New South Wales, Narrabri, New South Wales, and Toowoomba and Kingaroy, Queensland. So traditionally cover crops are used in the northern region. There are of course a multitude of these, but typical examples of grass species would be sorghum, millet, and Sudan grass. With respect to legumes, it's usually cowpea and lablab, although there are other inclusions as well. And of course, your brassica species in the form of forage brassicas. Unfortunately, in the south, we are not so lucky to get summer rainfall as much, at least traditionally. So we depend on a winter cover crop regime, mainly dominated by pasture species like clovers and annual medics, perennial legumes like lucerne, perennial grasses such as phalaris, and pulses such as vetch and field pea. But one thing to consider is that over the last few years, the rainfall patterns in the southern zone has also changed drastically. So for instance, in Wagga, the rainfall over the early summer season has been quite drastically enhanced compared to the rest of the year. And it's similar to what we see in Narrabri, but in Kingaroy, an example here, you can see that it's mainly dominated late summer, early autumn. So it makes a lot more sense where we start to investigate the inclusion of cover crops both for winter and summer in our region. So the soil types of the 3 sites that we picked are quite similar between Wagga and Kingaroy, being a red candesol, and in Narrabri it's a grey bertusol. So the winter cover crop species that we picked for our studies ranged many in numbers. So initially we started with 7 monocultures of covers and 9 mixtures, so both binary and tertiary cover crop mixes. What we did at first is screen them based on their ability to provide initial early season ground cover. To do this, we looked at NDVI, crop light interception, and of course crop and weed biomass. So the common cover crops we used for these were lupin, sardella, vetch, field pea, grazing oats, forage radish, and bicerola. So from our initial studies, we found that in Wagga, because of a very dry season, that early season ground cover was quite bad, especially at the 60-day mark. But as the time progresses, we only managed to get about 45% maximum ground cover, even with a grass species like oats. But in comparison in Toowoomba, where we had more irrigation available at the site, we were able to get much higher ground cover in things like radish and oats and vetch. So based on this ranking, we went through and produced a list of species for later studies. But we also looked at different multi-species mixtures that could be used. So in general, inclusion of a high biomass producing cover crop in a mixture produces more ground cover. So this is evident in Wagga, but in Toowoomba, you can see that the effect is much more prolific. So since we had 3 sites to look at, today I'll present data that we got from the cover crop trials done at Kingaroy, because the ones in Wagga Wagga actually did not produce very great results considering it was a dry season. So the species that we selected for this exercise were lupins, field pea, grazing oats, vetch, and tillage radish. And you can see that the canopy light interception was not so great for the lupins, which we kind of used as a negative control. We also used a fallow control in these experiments, but you can see over the growing season at 40, 60, and 80 days, the growth was quite prolific for the tillage radish and grazing oats. And the tillage radish actually produced nearly 80% of its ground cover within the first 40 days. What they've done in Kingaroy this time, because there's no natural weed seed bank that was uniform across the site, we've used mimic weeds. So here we've had Italian ryegrass and mustard, oriental mustard, used as a broadleaf mimic. So we'll see in a second how that panned out. So with respect to biomass, as we see with the ground cover, tillage radish definitely performed better than all the others over time. And if you then looked at the ryegrass population, sorry, the biomass, you see that tillage radish suppress the biomass accumulation of annual ryegrass significantly. And the same was for the pulses too. But Albus lupin, being very slow growing, produced the least amount of biomass, sorry, the highest amount of annual ryegrass biomass. So the mimic species we used as a broadleaf oriental mustard was not a great choice in the aspect that it was a very prolific weed mimic. But at the same time, it shows us that if we have an exceptionally vigorous weed, most of these treatments would not be able to suppress its growth. So what we also looked at was the impact of multi-species mixtures on the accumulation of biomass in weed mimics. So again, the ryegrass is in the green, mustard in the yellow. So we have a tillage radish and field pea treatment here. So the only treatment that actually significantly dropped the mustard population was tillage radish. This data needs to be taken with a grain of salt because it's actually quite difficult to identify between radish and mustard. So there could have been slight exaggeration of the result there. But in general, the ryegrass population, the biomass actually was suppressed significantly as we increased the tillage radish percentage relative to the recommended sowing rates. Same was true for grazing oats and purple vetch treatment, and also the grazing oats and field pea. But as you can see, with field pea alone and purple vetch alone, the ryegrass biomass was actually higher than the other combinations. At the Kingaroy site, unfortunately, the soil is under the surface quite a rocky situation. So although we didn't have much biomass produced in Wagga, we used the soil moisture measurements from there as an indicator of how things were going. So you can see over time that the soil moisture levels reduced at 20 centimeters below the surface. So we used a TDR probe for these measurements. Whereas in 70 centimeter depth, there was not much of a difference, meaning our crops actually had not established very well. But this gave us an opportunity to try out doing soil moisture measurements. This is our first attempt at it, and definitely gave us a lot of pointers as to how to proceed for next year's. And in fact, the 2020 growing season is proving very well, where we have increased the number of replicates to reduce the noise that we see in these graphs. So similarly for summer cover crops, the preliminary experiments contained 11 monocultures and 2 binary mixtures. And again, we did a heat map where we could figure out which ones were growing much more rapid compared to others. So generally speaking, the sorghums, the usual culprits like millet and sorghum, cowpea, and Sudan grass produced a lot more ground cover early on, both in Narrabri and in Toowoomba. And we use these for experiments later on. So here's an example of the canopy blight interception from this year's, in 2019 trials conducted in Kingaroy. So we had buckwheat, we had cowpeas, we had a fallow treatment, French millet, Japanese millet, lab, and sorghum, all of which accumulated, produced early season ground cover with the exception of lab. We also looked at the two mixtures, French millet, cowpea, and French millet and lab. So as you can see, as monocultures, cowpea and lab produced less ground cover early on. But as soon as you mixed in millet, even at a lower percentage, the early season ground cover was established. The story was quite similar with the biomass, where we see sorghum produced the most, over 12 ton, whereas the others were ranging between 6 and 10. In this case, the wheat mimics chosen by the group were lucerne as a dicot indicator and Rhodes grass as a grass species. So that's relative to the fallow. So you can see the fallow had the most amount of biomass for lucerne, but all the treatments managed to reduce that biomass significantly. And the same was true for Rhodes grass, with the exception of lab, which didn't produce early season ground cover. As soil moisture measurements were not possible there, giving you an example of the same experiment performed at Narrabri, where we have a few representative species here. So we have French millet, sorghum, cowpea, and buckwheat. So the crops here were terminated at 40 days after emergence and 80 days after emergence. So you can see at early termination, so the orange bars are at a soil moisture at a depth of 70 centimeters. The blue lines are soil moisture at 20 centimeters below the surface. So definitely the late termination had an impact on conserving soil moisture in most of these cases, but the signal was quite noisy, possibly because of the irrigation events that took place during this event. But this year's trials will definitely have more replication of the soil moisture, and we are hoping to publish these results in the coming year. So in conclusion, the multi-site field experiments that we did over winter and summer growing seasons enabled us to identify cover crops that were better suited for the northern region to suppress weeds. The crop biomass at the early and late vegetative growth stages and light interception at the early vegetative growth stages did contribute to the reduction in weed biomass accumulation. And we came to this conclusion based on a linear regression model. However, as you know, most of the work that we did over the last 2 years was under drought conditions, 2020 being the best coming up season with more representative growth parameters. So we're looking forward to the results from this coming year. So the other aspect of this is we've been looking at this in-season weed suppression, but the crop residue impact on weeds is quite an important consideration. So for that, we are looking at the crop residues that are produced under field conditions and their impact on suppressing weed species. So we are doing a few laboratory assessments on that and a few glasshouse studies. We are also collecting the tissue material for these suppressive crops, and we are going to look at the secondary metabolites produced either in the crop residues themselves or the rhizosphere through root exudations. With that, I would like to thank for the opportunity to have this presentation, give you the presentation. And I would also like to acknowledge funding provided by the GRDC project and the collaborating institutions, both QDAF and University of Sydney, all the technical staff and students, and the Graham Center for providing conference support. Thank you.
All right, that's good, Sal.
Thank you, Michael.
So as you can see, there's a couple of questions waiting there for you in the chat room.
I'll just stop sharing so I can see it.
Yep.
There it is. So the first question is actually from Ali, looking at mixtures where then, so evidence is mounting that mixtures are no good when it comes to weed suppression. What's your verdict? Ali, that's actually a really good question. What we are seeing in a really good growing season like 2020 is that some mixtures we are looking at, specifically the FLP and grazing oats treatments, are having a lot more weeds present compared to the others. So I think that's mainly due to the growth characteristics, where you have a late maturing plant like FLP that gives the opportunity to have a lot more ground cover available to weeds. So I agree with that, but I think it mainly comes down to the decisions that we need to make as what the weed species that you are combating, because we are looking at some of these, the data from Kingaroy, where we have the dicot and monocot species, and we are seeing a clear difference as to the specificity of suppression towards them. So that I hope will answer your question. And Mark has a question here about any thought of having both an unsprayed and sprayed fallow as comparative standards. In practice, the competitive standard is a sprayed fallow. That's very true, Mark. In the initial stages, we actually couldn't facilitate having all this, considering the number of treatments that were involved. But that's definitely the case going to be for the fall, for the preceding years. So we will definitely look at that. Ali, did you measure the actual emergence of different species? I'm assuming you're talking about the crop species. Yep, we definitely looked at the individual species in mixtures. We also looked at the emergence of the weed mimics in these treatments to make sure that we actually had uniform emergence, and they were all very uniform. And in the mixtures, the percentages matched the recommended rates that were given. So if we had a 50-50 split between two species, we definitely had the emergence of 50% of each of those components. A question from John Cameron. Pathway to adoption. This is always a good question, John. So this is a change in farming system with substantive impacts way past weeds alone. What needs to happen to enable that case to be made? John, that's one reason the variation is looking at other aspects of using the cover crops, because although this project was targeted towards managing weeds, it's become a bit more than that, where you're looking at the impact on soil moisture, and also the impact on the finances in an enterprise perspective. So the final year of the project is earmarked to look at the financial benefit to the growers and to see what is the long-term cost and the benefit towards them, particularly if you have a resistant weed present. I hope that answers your question.
Can I just add a little bit more around that, please, Sal?
Sure, sure, Michael.
I think it's fair to say that weed control is a secondary part of cover crops. It's really about soil surface coverage and residue retention, and that weed control is a secondary benefit.
That's correct. Any other questions?
I've got a question for you, Sal.
Sure, Michael.
Yeah, I was just wondering whether you wanted to elaborate a little bit more about how you chose the termination times.
Yeah, that's a good question. Thanks, Michael. So we were aiming for 2 time points. First being late vegetative, where we would have the maximum biomass accumulated, and also the highest amount of ground cover. And the second one being at maturity, because that changes between species, the whole dynamics between the amount of foliar content and grain and all that. So those are the 2 main reasons. And if you actually coming from the narrowbri data, it's quite interesting to see as soon as you switch over from that vegetative to reproduction stage, the amount of soil water use just goes crazy after that. So it's quite important we understand the difference, and that's the reason we picked those 2 time points. Any other questions? Looks like that's it, Michael.
Yeah, all right. Excellent, Sal. All right.
Thanks very much, Michael.
Thank you very
Thanks
much
very
for your presentation. Thanks for both presenters, and yeah, consider yourselves receiving a sitting ovation. And yeah, thank you all for joining, and look forward to next week's presentations.
Great. Thank you.
Thanks, guys.